Today, we're jumping into a core concept in biochemistry, enzyme kinetics, and we're going to make the famous Michaelis-Menten model super simple to understand.
Think about it. Enzymes are running the show inside every single one of your cells. They control the speed of everything, from digesting your lunch to copying your DNA. They're the tiny engines that make life happen.
Okay, so if they're engines, how do we measure their horsepower? How fast can they actually go? Well, that's exactly what enzyme kinetics is all about. And the Michaelis-Menten model, that's the language we use to understand it.
All right, let's get into the nitty-gritty. An enzyme is basically a molecular catalyst. Its whole job is to make a reaction go way, way faster than it normally would. And here's the cool part. It doesn't get used up in the process.
It's this really elegant three-step dance. First, the substrate, that's the molecule the enzyme works on, binds to a special spot called the active site. They link up to form what's called an enzyme-substrate complex. Then, boom, the reaction happens, a product gets released, and the enzyme is totally unchanged, ready to do it all over again.
So if we want to know how efficient an enzyme is, we need to measure its speed, right? We call that the reaction rate, and the classic way to do that is just to see what happens to that speed as we keep adding more and more substrate into the mix.
And when you plot that out, you get this classic curve. See how it works? On the bottom axis, you've got your substrate concentration. As you add more, the reaction rate on the vertical axis just shoots up at first. But then it starts to level off, and eventually it just hits a ceiling. It plateaus. So what's going on there?
That ceiling, that plateau, right there, that's what we call Vmax. It is the enzyme's absolute pedal-to-the-metal top speed. It's a maximum velocity this reaction can possibly hit.
The key word here is saturation. Vmax happens when literally every single active site on every enzyme molecule is busy. They're all occupied. The enzymes are working flat out, as fast as they can. You can throw more substrate at them, but it won't make a difference because there's just nowhere for it to bind. Okay, so Vmax tells us about max speed, but that's only half the story. The other piece of the puzzle is a value called Km, and it tells us something just as important, an enzyme's affinity for its substrate.
So how do we find this Km on our graph? It's actually super simple. You just find the Vmax at the top, cut that value in half, and then look across to see what substrate concentration gets you to that half-speed point. And that concentration, that's your Km, the Michaelis constant.
Now, listen up, because this is probably the most important thing to get about Km. It has an inverse relationship with affinity. So, a low Km means the enzyme has a high affinity for its substrate. It grabs on tight and doesn't need a lot of substrate around to get going. But a high Km, that means low affinity. The enzyme needs a whole lot of substrate around before it'll really get to work.
And this is where it all comes together in one beautiful formula, the Michaelis-Menten equation. It ties everything together, the reaction rate, V, the max speed, Vmax, the affinity, Km, and the substrate concentration. It's the foundational equation for modeling how pretty much all enzymes behave.
So why does all this matter so much? Well, because sometimes we want to slow enzymes down. And understanding how inhibitors work, these are molecules that basically hit the brakes, is absolutely critical in biology and especially in medicine.
All right, there are three main types of inhibitors to know. First up, you've got competitive inhibitors. They do exactly what it sounds like. They compete with the substrate for the same active site. This makes the Km go up. It seems like the enzyme has lower affinity, but if you add enough substrate, you can still reach the same Vmax. Next, we have non-competitive inhibitors. These guys don't bind at the active site. They bind somewhere else and change the enzyme's shape. This messes up its function, so the Vmax goes down. But since it doesn't affect the active site directly, the Km stays the same. And finally, there's a weird one, uncompetitive inhibition. This inhibitor only binds after the substrate is already locked in. It essentially traps it, which means it ends up lowering both Vmax and Km.
And I have to stress, this isn't just some abstract theory you learn in a textbook. Understanding enzyme kinetics is how we design many of the drugs we rely on every single day.
Seriously, so many modern drugs are just really, really good enzyme inhibitors. Think about statins for cholesterol or certain antiviral drugs. They're all designed based on kinetic data. Scientists use this information to create molecules that can target and block a specific enzyme involved in a disease. It's how we turn basic biochemistry into actual life-saving medicine.
Okay,
Let's wrap this up. What are the big things you need to remember from all of this?
It really boils down to two things. Vmax is the enzyme's top speed, its maximum capacity. And Km tells you all about its affinity for the substrate, how well it binds. You put those two together, and you get this incredible snapshot of how an enzyme works. And that knowledge, well, it's fundamental to everything from basic biology to cutting-edge drug design. So in the end, it's Km and Vmax that really define how an enzyme does its job. Understanding this stuff is absolutely fundamental, whether you're in biology or bioinformatics. If you want more clear scientific breakdowns like this one, make sure you subscribe to Bioinfo Bytes.